Okay, so yeah, so we're gonna have a look at the biology 2021 module one, right? So it starts off with this Escherichia coli of a magnification there, right? What we need to do is from the electron micrograph produce a diagram, right? So I already stated that we know can't really do the diagram properly on this digital apparatus that I'm using, but we know how to calculate the magnification. The magnification is I over a, lovely, right? So you need to carry. Please, I beg, carry rulers into the examinations as well as the calculators, because if they give you like three join and a one chi-square test, I told you so, right? Because chi-square test is very rare, right? But notice that chi-square test really came what 2017 and that's it. I don't see any more chi-square tests, right? But carry calculator and your ruler so you can have a look at your biological drawings and well, you calculate on your ruler rather. Alright, so list three similarities between a typical animal and typical plant cell. What are the similarities now between animal and plant cells? See, biology students could handle this. Go ahead. Or somebody said we're definitely getting Casper. Alright. Interesting. What's the similarities now? Both have nuclei, right? What else?
So they both have a plasma membrane.
I mean, yes. Plasma membrane. This is a really easy paper, right? Judging by the module one, it's a very easy paper. What else? DNA cereals chromatin. What else? As membrane bone organelles as well. Okay, moving on. Outline the function of any two structures found only in plant cells. Cell wall. What is the function of the cell wall?
So control what enters and exits.
So not only does the cell wall does that, but the cell membrane helps with that as well. Alright. And also for general structural integrity. And protection. Alright. Why my T is not in my integrity? Alright. We have chloroplast. What is the function of chloroplast?
So contains chlorophyll for photosynthesis.
Right, it's a site of photosynthesis. Alright. What is this thing? Why is it on my screen and why it is go? Alright. So it's a site of photosynthesis. Alright. Just basically just outline the function of those things.
Converts light energy to chemical energy for growth.
Yes, which is photosynthesis. Lovely. Alright, so any way you want to put it, it's still going to be the site of photosynthesis. Alright? Lovely. Cellulose and starch, which consists of amylose and amylopectin, are two important polysaccharides found in plants. Discuss three differences in the structure of starch and cellulose. Let's start with starch. What structure of starch that we see? List them now because we're supposed to have all of them on lock and key. Alright? Go ahead.
So we're going to talk about like the alpha 1 for glycosidic bond.
Contains lovely alpha glucose. Right? Usually within the alpha, oh, that's not how we spell alpha. Alpha 1 for glycosidic linkages. right? What else do we have? It's a mixture.
Cause in helix shape and compacts together as it is soluble in water and does not affect the water potential of cells.
Okay, it's soluble, right? Helical shapes in shape, all right? And then what is the percentage generally? It's usually 10 to 30%. 30%. Amylose, and then the other is usually the rest of the percentage of for amylopectin. All right. Cellulose, no. I will just put in structure so you guys can make your own distinctions, right? Cellulose, no. What happens now?
Uses beta glucose units.
Okay, all right. Somebody else was saying something as well, so somebody else I don't know. So remember that these have 1,4 glycosidic linkages and amylopectin has some 1,6 glycosidic linkages now.
Amylopectin has both alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bond.
Okay, it's not as much as glycogen. All right, lovely. So beta glucose now uses 1,4, right? We know that, right? So know that every, let me asterisk, every other glucose molecule is flipped. And that allows for hydrogen bonding between the strands. And that allows for the formation of microtubules. Well, microfibers. Microtubules, microfibers, whatever you want to call them. Right? And then those would now compact into strong isomers. Not strong isomers, strong poly saccharide chains. Right? What else we could state? It is insoluble. Insoluble. Alright? So we'll talk about those type of things. You guys should be able to find three differences there. Right? They're sheet-like in structure. They form sheets. Right? So it's really sheets. And then the sheets will wrap now into microtubules. Right? Okay, or microfibers. Alright, that's it for that question for module 1.